All right, well, let me start by saying good evening to everybody who's on this meeting. I'm Stephanie Burton. I am the immediate past president of the Royal Society of South Africa, and therefore still on the council, and that's my role tonight in hosting this lecture on behalf of the Royal Society of South Africa. I am going to make one or two general announcements in a moment, and then after that I will introduce our speaker, and we will be able to listen to what I think promises to be a very fascinating talk. So allow me please to just share my screen and just talk very briefly about, there we go. We will have our talk in a moment, but before I begin that, let me just say a little about the Royal Society of South Africa, which we know is one of the oldest, it is the oldest scientific organization in South Africa, and it is open to anyone who has an interest in science. And if you are a member, you can, of course, join into talks like this. There is access to our journal, The Transactions of the Royal Society of South Africa, and there are invitations to various meetings and congresses, and all our members are welcome to attend all of those, and they will be notified about them. There is an application form on our website, and there is also a special rate for students, which I think we very much encourage. Sorry, wrong way. We are also affiliated, at least in partnership with the South African Academy of Science of South Africa, which is our officially recognized science academy, which was founded only in 2001, but is the official evidence-based scientific advisor for the national government and many stakeholders. And this series of talks is run in collaboration between the Royal Society of South Africa and the South Africa's Academy of Science. So again, if you are interested in more about the Academy, there is our website, and you are, of course, welcome to take a look at that. Just to mention that we are going to have this talk tonight, and we have these talks regularly. So the future talks that we will be having in September and October will be rather different. The first one on physics with Dr. Isaac Nape, and in October, Dr. Dudu Arin, who is an engineer in materials science. So please do keep a lookout for those. We will be notifying our members about them, and you will be welcome to listen to those as well. Oh, these slides, right. So let me now introduce our speaker, Professor Gordon Brown. Gordon is at the moment based in Exeter. He's the director of the MRC Centre for Microbial. I'm going to stop this sharing because it's on an automatic timing base. Sorry. There we go. Gordon is the director of the MRC Centre for Medical Mycology at the University of Exeter, and he's director of the Africa Medical Mycology Unit at the University of Cape Town. He has a BSc in microbiology and genetics at the University of Wits, and then he did a PhD at the University of Cape Town, and his PhD supervisor is on this call with us, Professor Jennifer Thompson. And then following that, he undertook a postdoc at University of Stellenbosch, and then moved to the UK to the University of Oxford. He returned to Cape Town in 2004 as the Wellcome Trust Senior Fellow and established his own group then. And in 2009, moved to Scotland to the University of Aberdeen, where he established the MRC Centre, of which he is currently director. And then in 2019, relocated to the University of Exeter. So Professor Brown has been around the environment, if you like, but he's going to speak tonight about his area of interest, which is understanding how our immune systems recognize and control infection. And he's called it the good, the bad, and the devastating. And I think that is going to make for a very interesting talk. So let me hand over to Professor Brown, and we look forward very much to what you're going to tell us this evening. Thank you.
Well, thank you very much. Can you hear me okay, Stephanie? Is it coming through?
We can hear you fine, yes. And we know that your screen is going to share successfully.
Yep.
There we go.
There we go. Right. Well, thank you very much.
Thank you.
Thank you very much. And thank you very much for the introduction. Yes, as you can hear from my accent, I'm a South African based here in the UK, but have very strong links still with Cape Town in South Africa. Most of my family is residing there. So what I really want to do today is to tell you about a focus of my entire career, which is really how our immune systems recognize and control infection. And this work started for me when I went from Cape Town to Oxford and made a discovery that really changed my career in terms of my interests. And what I want to do today really is to explain to you and to show to you why I find immune recognition so interesting, and some of the remarkable discoveries that we've made over the last 20 years or so. So what I thought I would do today is to really just give you an overview of the area and field, and then focus in specifically into the group of receptors, immune receptors, that I'm interested in. And at the end of my talk, I'm going to focus on a really recent discovery that we've made, which was published last week, actually, which has really changed our understanding of how the immune system recognition of pathogens and response to pathogens can be very important in terms of the balance between protecting against infection, but also driving the development of autoimmune disease. And I'll get to that later. So let me first start by changing my slide, which is that one, which is to really set the scene a little bit. So what you see in front of you is a picture of an immune cell that you can see over here, and it's recognizing and responding to this microbe down here. And you can see very clearly that this immune cell is recognizing it, and it's responding to it, and it's sending out this process that would ultimately engulf and kill this pathogen. Now, the bit that really interests me is how, if you've never seen a pathogen before, how does your immune system recognize infection? What is it about your immune system that enables it to see this? It must have some sort of mechanism. And this recognition is absolutely crucial for your immune system. So because this recognition, characterized in this cartoon here, really leads to the ingestion of microbes, which kills them. But importantly, this recognition also drives very important inflammatory responses that recruits other immune cells to the site of infection to enable it to control the infection. But at the same time, send off information to your adaptive immune system, your T cells and B cells, and allowing them to be instructed about the nature of this pathogen and to mount an appropriate response. Now, when I entered this field in the late 90s, we knew that immune cells could recognize pathogens, and we knew that they could trigger these responses. But the really big question that existed in the field at this time was, how does this occur? What is the recognition system that occurs on immune cells that enables this really key process to be initiated? And the real big breakthrough that came from this was a discovery in fruit flies in Drosophila by Bruno Lemaitre and Jules Hoffman, shown over here. And what they discovered in Drosophila was that if you knocked out a protein called toll, that these flies became very susceptible to a fungal infection. And this iconic picture from the period really shows a fruit fly that's died from a fungal infection. This is not a normal state of affairs. What you see here is fungal hymphae absolutely bristling out of these flies. And this was really the fundamental discovery that led very rapidly to a discovery of similar proteins in humans, the toll-like proteins, done by Ruslan Metsitov and the late Charlie Janeway, and then also independently by Bruce Beutler. And in fact, these individuals, Jules Hoffman and Bruce Beutler, were awarded the Nobel Prize in 2011 for this discovery. So it was such a fundamental discovery. And the reason why it was so fundamental was because this discovery of the toll-like receptor, which is shown schematically here, really allowed us to understand how our immune cells could sense pathogens at the cell surface. So these are cell surface receptors that then, upon sensing the pathogen, trigger the intracellular signaling that initiates this really complete induction of the immune response. So this really was a really paradigm shift in our understanding. It allowed us to understand how the immune system is able to recognize and respond to pathogens. Now, the key here is that this recognition system is of commonly conserved components in microbes, so what have been termed pathogen-associated molecular patterns. And a common one of this is endotoxin or lipopolysaccharide found in gram-negative bacteria, which is a very strong agonist or very strongly recognized by the toll-like receptors. So the way our immune system works then is that it has these genetically encoded receptors, so that enable it to directly recognize these conserved structures. And then these receptors, through this recognition, then trigger these intracellular signaling cascades that drive the development of this very key immune responses. And I should point out, and I'll make this statement quite early on, actually, that your entire immune function is critically dependent on these receptors, because we now know that defects in these receptors or the intracellular signaling pathways make you very, very susceptible to infections with a variety of microbes. So this award of the Nobel Prize really started a cascade of events of people looking for new receptors and other receptors other than the toll receptors. And this is where I came in. So I started in this field just after this discovery was made. And my contribution to this area, along with Simon Gordon in Oxford, in actual fact, was the discovery of another class of receptors, or called pattern recognition receptors, because they recognize these molecular patterns, as I've just said. And this other class of receptors was called the C-type lectin receptors. Very similar to the toll-like receptors in that they have a domain that enables them to recognize conserved structures on microbes, but also intracellular signaling domains that are able to drive these immune responses. And then subsequent to this discovery, there's been discoveries of other receptors, including ones that actually exist inside cells that allow them to detect viral infections, for example. Now, since this discovery, my entire career really has been focused on understanding the roles and functions of these C-type lectin receptors in immunity. And like the toll-like receptors, try and understand what it is that they do to the immune cell that they are expressed on, and how their signaling domains then drive all of these cellular responses. And I'll give you some examples of some of that in a moment. But first, let me just tell you a little bit about C-type lectin receptors. So, and actually, as it turns out, the C-type lectin receptor family is an enormous family of proteins, all characterized by these brown Pac-Man-looking structures. These are the C-type lectin domains. In actual fact, this is a bit of a misnomer. C-type lectin comes from the original discovery of these molecules as calcium requiring sugar binders, in other words, C-type lectins. It turns out that this C-type lectin is a structural domain that's found in a really wide variety of proteins, as you can see here. Over 10,000 proteins in the metazoa have been identified to have C-type lectin domains, and they serve a whole variety of functions. Now, I say that C-type lectin domains as a name is a misnomer. That's because these molecules, in actual fact, don't all require calcium, and many of them have a very diverse range of ligands they can recognize in addition to sugar, things like proteins and lipids, etc. What's interesting about these proteins is that they are a relatively unstudied group of molecules, and they are absolutely essential for our existence. They play roles spanning every aspect of multicellular existence, from allowing cells to move around in your tissues in your bodies. Proteins such as this protein over here that I'm highlighting, the surfactant proteins, allow you to breathe. We've got proteins such as tetranectin that have been indicated in longevity in humans. So really a huge variety of activities and functions that these proteins play in controlling our existence in multiple levels. But the group of molecules that I'm particularly interested in is this small group down at the bottom here, these small group of receptors, particularly the ones that have this single C-type lectin domain, and then are either associated with or directly hold a transmembrane domain that allows the cell to trigger intracellular signaling. And the receptor that I discovered that really led to this was this receptor called Dectin-1. So let me introduce you to Dectin-1, because we've done a lot of work on this molecule over the last few years. So this is Dectin-1 here. As I said, it's got a binding domain, a C-type lectin domain outside the cell, and a tail on the inside of the cell that allows it to trigger intracellular signaling. The discovery of this molecule led to the discovery of a novel signaling pathway in immune cells. This pathway is indicated by SYK and CAR9. It's not really that important. It's just the signaling pathway. And we've learned that there's many other pathways being induced by this receptor that allows it to influence the behavior of immune cells. What is particularly fascinating to me is that a molecule, this is a very small 30 kilodalton size protein, so a very small little protein in immune cells, is that it has a multiple of functions. So it can recognize pathogens through its ligand binding domain. It can cause these pathogens to be taken up by immune cells. It can trigger the immune cells to trigger a whole variety of inflammatory responses, inducing inflammatory proteins such as cytokines. It can induce inflammatory lipids. And it can drive all of those responses I showed you in my cartoon previously. It can instruct the adaptive immune system to respond appropriately. Now, what was very interesting about this discovery, which will become relevant in a moment, is that this receptor we discovered recognizes this conserved structure called beta-glucan, which is a sugar, but is found very widely in fungal microorganisms. And in actual fact, this really pushed me my career towards studying the role of these immune receptors in the control of fungal infection, which we'll talk about in a moment. Now, what's particularly interesting about this receptor, as I said, is its multitude of functions. And what I'd like to do is I like to show these movies that I have to the right here, which really highlights to me, and hopefully to you, just how amazing these molecules are. So what you have here on the top of the top movie is a immune cell that doesn't express this receptor. So this is an immune cell that we have in the lab. And you can see that when we add in our fungal particles that express this ligand to these cells, you can see that the cells are clearly in contact with these particles, but are not responding in any way. Now, these particles, if you take these cells and you now express this receptor, so this is the same cell, right? But now all the difference between this cell and this cell is that this cell is now expressing Dectin-1. There is, to me, an absolutely amazing difference in the ability of these cells to recognize and respond to these particles. And you can see here that this immune cell is being able to recognize and ingest these fungal particles. And this recognition event and binding event then triggers all of these cellular responses. It's absolutely key, as it turns out, for the control of fungal infections. So what we've gone on to show over the years in a variety of different contexts, both in humans and using our mouse models, is that this receptor is absolutely essential for controlling antifungal immunity. And here's one example. So this is a mouse model where we've actually knocked out this receptor on all the cells in the mouse's immune system. And you can see when you infect mice now with a fungal pathogen, in this case, we've used Candida albicans as our model. You can see that the mice that possess Dectin-1, that's shown here in this black square, they are able to resist, looking at survival, resist this infection over time. But if you take the same mice now, but knock out the Dectin-1 pathway, you can see that these mice are very much more susceptible to the disease, and they succumb to this infection much more quickly. What we've got on to also show and in recent years that, of course, this receptor triggers all of these responses. I showed you that in the last slide, but also instructs the adaptive immune system to respond appropriately. And we've shown through a variety of studies that this receptor is able to induce specific types of adaptive immunity that's really important for controlling fungal infections. Now, I'm sure from the recent inflammation that we've seen in COVID and other more recent examples, you're aware of how important it is that to have a functioning immune system. But what's been particularly interesting about studying of receptors such as Dectin-1 is this ability to instruct the adaptive immune system to drive very specific immune responses that are protective for controlling fungal infections. And perhaps the most interesting of these was this type of adaptive immune response called a Th17 response. This is an adaptive immune response where there's a cytokine that's called IL-17. It's not relevant. But what's particularly interesting about this Th17 response is that it's been shown fundamentally to be important for controlling fungal infections at your mucosa. And in fact, if you look at humans and humans who have genetic predisposition to fungal infections, it's most commonly defects in this arm of the immune response that causes susceptibility to the disease. And it's been postulated that the evolution of this adaptive immune response through these sort of receptors has actually been evolved to deal with fungal infections. So really important that these receptors exist and allow us to drive appropriate immune responses that control infections. And since this initial discovery, there's been going on the way. There's been a very large resurgent work from my lab and many other labs have identified a whole variety of other receptors that are able to recognize and respond to fungi. And all of these receptors signal through the similar pathway and induce these immune responses that are really key for controlling human and mouse antifungal immunity. I'm just going to take a moment, really, because since this discovery of Dectin-1 in my career, that really pushed me to move into the field of medical mycology, the study of human fungal infections. And I want to just take a moment to step away from my topic, just to highlight to you just why studying or being aware of human fungal infections is so important. Because of all the infectious diseases, human fungal infections are the least recognized and understood. And I'm sure all of you are aware, of course, of TB, and you're all aware of malaria. But I'm sure that not many of you are aware of the huge impact that fungal infections have on human mortality and morbidity. So amazingly, more than a quarter of the world's population, including, I would suspect most of you listening, have suffered from one or more fungal infections in your lifetime. Most of these fungal infections occur are not really lethal infections. They occur in your skin and nails and mucosal tissues. So things like dandruff, athlete's foot, or thrush are all things that I'm sure you're aware of, all caused by fungal infections. But in the last few decades, the last 30 years or so, there's been a huge rise in the number of life-threatening infections. These are invasive infections that occur in people. And what's particularly scary about these infections is that they have very high mortality rates, often exceeding 50%, and in some cases up to 80% of people who get these infections die. And so what this means is that there are probably the same number of deaths caused by fungal infections as they are caused by tuberculosis. So about 1.8 to 2 million deaths every year are caused by fungal infections. And that's 3 or 4 times more than that's caused by malaria. So a huge burden of infection, not widely recognized. Yet, as I say, they are creating a real issue for human morbidity and mortality. So why is it only over the last 30 years that we've seen this tremendous rise in fungal infections? Well, it's primarily because our immune systems have evolved to deal with fungal infections, and we are very effective at doing it. But over the last 30 years or so, with the rise of HIV/AIDS and the advent of modern medical interventions, even simple things like catheterization, both intravenous and elsewhere, have overcome many of the barriers that our immune systems use to control these infections, and have led to this huge increase in mortality rates. And what's particularly scary about this is that most of these deaths occur in low and middle income regions of the world. So one very typical one, for example, in sub-Saharan Africa, including South Africa, is cryptococcal meningitis occurring in HIV-infected patients, and it's probably the second leading cause of infectious death after tuberculosis in HIV patients. But in actual fact, we have very little idea of the actual numbers because we have very little epidemiology of these infections. And what we need to actually tackle these infections, we need better drugs. So unlike bacterial diseases, we've only got 4 classes of drugs. And like bacterial infections, there's a huge rise in antifungal drug resistance. We need better diagnostics for these infections. So most of the fungal infections we don't have diagnostics for, or diagnostics that are not at point of care. So you're all familiar with lateral flow assays that was very useful in detecting COVID. For most fungal infections, there's no lateral flow assays, although for Cryptococcus, there's just been one released recently. And perhaps this alone is responsible for many of these deaths, because by the time clinicians diagnose fungal infections, even if they're aware of fungal infections, it's too late, and the infection is too deep-seated and they die. And we also need a better understanding of the host and pathogen interaction, because as I mentioned, it's the immune system that's key, and it's the compromised immune systems that lead to systemic infections. And it's hoped that understanding how the immune system recognizes and responds to fungal pathogens will lead to sort of adjunctive immunotherapeutic approaches. And there's examples of that already occurring, where our understanding of the underlying immunology has led to cures that can help patients, specifically, for example, patients with cryptococcal disease. But because these infections are so poorly studied, and because we have so little understanding of these infections, there's been a lack of interest. And this has led to a huge lack of clinical and basic capacity in tackling these diseases. I mean, so rife that some countries have no research at all. And most clinicians, for example, have very little comprehension of the fact that fungal infections occur. And this is something that we are really passionate about here at the MRC Center where I'm based. And one of the ways we're trying to deal with this is to increase capacity in basic clinical sciences. And so in the introduction, it was mentioned that I'm the director of the Africa unit in Cape Town. And this is part of our global outreach activities to try and increase awareness and capacity in low and middle income regions of the world, which I've already told you have the highest burden of fungal diseases. So we created the Africa unit in Cape Town. This unit really serves South Africa, but is aimed at serving the whole of Africa. And we often hold training and capacity building courses in Cape Town that really reach out to the rest of Africa. And the focus of this unit is really around research and training and capacity building. And we're just about to launch a new unit in Brazil. So this is launching next month, and then we will open up one in Asia, Southeast Asia. And the purpose of these units is to really, as I say, try and build capacity and create an international network of scientists and clinicians working in medical mycology to try and capitalize on joint experience and joint knowledge, very similar to what the TB community and HIV community have done very successfully recently. So really building capacity in this field is really important. Okay, so that was a slight deviation, and I want to get back to my talk now and get back onto the topic of discussion. So I mentioned to you that we've been studying C-type lectins in the context of fungi, and a lot of my work has been really revolved around understanding how this all works and how the immune system recognizes and responds to these pathogens. But since this discovery, we've also realized that these molecules, these C-type lectin receptors that we're so interested in, actually are involved in the recognition of a whole variety of other pathogens. They can recognize mycobacteria, for example. They can recognize viruses, helminths, and pathogens. So these receptors, although critical for controlling fungal infection, are also involved in the control of many other infectious diseases, both in a positive manner. So, for example, I've already shown you that the Dectin-1 responses in response to fungi is protective, but also in a negative manner. So a very good example of that negative manner is this receptor is called CLEC5A. This recognizes dengue virus, which I'm sure you've heard about. So it recognizes dengue virus. And in fact, the pathology that comes from dengue virus is actually mediated by the immune activation through this receptor. So this is the good and the bad. They both play protective and non-protective roles in immunity. And some of these responses can be devastating. So the pathology driven by dengue virus can be lethal, as I'm sure you're aware. Now, in addition to recognizing pathogens, many of these receptors also recognize damage to self. So if you, for example, have an injury, you get burnt, you get a cut, your cells are damaged. And a lot of these, what I told you were pattern recognition receptors, not only have evolved to recognize microbial patterns, but also self patterns, so that it can alert the immune system to damage to your bodies and the integrity of self. And a very good example of that is a molecule called DNGR1, Danger1 as it's called, or its real name is CLEC9A. And what that recognizes is a structural component of all cells called F-actin, which I'm sure you're aware gives mammalian cells its structure. And so if the cell bursts open, this F-actin is released. This allows the danger to recognize that. And that's really important for controlling immune function in response to things like tumors and viruses and other aspects. Now, most of my career has been focused on studying these receptors and their ability to activate the immune system. So I mentioned to you that this pathway that Dectin-1 is recognizing that triggers an intercellular signaling system that we very well understand now, and that then leads to cellular activation. And that activation is really cool because it drives immune responses that allows us to combat infection, and in some cases, I've just said, is detrimental because it can drive pathology. But there's another class of these receptors, which is not found in other pattern recognition receptors, are ones which have a pathway that actually dampens cellular activation. So this SHIP1 kinase is phosphatases, as shown in this DCIR here. Now, these receptors, of which there are a lot, and I'm only showing you one, but actually people don't know what they do, because the difficulty in studying these receptors is that, in contrast to the activation receptors, where you trigger the receptor, you get a cellular response that's easy to measure. When you trigger these receptors, you're blocking something. And what that something is that's being blocked is not known. And that has made these receptors not very well understood, and is why we have very little knowledge about the plethora of these inhibitory receptors and what they can do in the control of immune function. Now, we know that they do play a role in the control of immune function, because on the 1 or 2 or 3 of them that have been studied, when you knock these out in mouse models, what you see is that these mice tend to develop autoimmune disease, and they develop quite severe autoimmune disease. So what I mean by autoimmune disease, I mean by there's a lack of endogenous or homotypic control of the immune system, and this leads to damage to self. So there's a damage to self occurring, which can be quite severe. And so this brings me on to the last part of my talk, the focus on this new discovery that we've made, which has really completely changed at least my understanding of how these receptors function, and really shows you how important these receptors can be in the balance between driving responses that are protective, for example, from infection, but also driving responses that are detrimental, like in the context of autoimmunity. And so the molecule that I want to introduce to you now is this molecule that we actually discovered many, many years ago, around about the same time as Dectin-1. It's very similar to Dectin-1 in terms of its structure, as you can see here, but unlike Dectin-1, it has a motif that is an inhibitory activation. It blocks inflammatory responses, and it doesn't activate. And so although we discovered this receptor at the same time as Dectin-1, so that was about 20 years ago, it's taken us 20 years to figure out what the hell this molecule is doing in the immune system. And so we did a whole variety of relatively artificial, you can see all the publications of relatively artificial studies looking at how this receptor functions. We could show, yes, it is able to trigger an inhibition of cellular responses, but we really didn't understand what it was recognizing, and we didn't really understand what the true immunological cellular response was being recognized by this and controlled by this inhibitory zone. So as I mentioned to you, these molecules, these inhibitory molecules, have been shown to play a role in controlling autoimmune disease. So our first forward step in our understanding of this molecule was when we looked at the role of this receptor in the context of arthritis, right, which is an autoimmune disease, as I'm sure you're aware. And there's a variety of mouse models of arthritis, and we explored the role of this receptor in a variety of models. I'm going to show you one. And this is a model of arthritis. I'm going to try and keep very superficial here in terms of detail, but some of it will be important. So basically, we have a mechanism for inducing the arthritis in the mice. And then we allow the arthritis to occur, and we can measure a variety of parameters. We can measure the clinical score in the animal. So, for example, we can measure joint inflammation in the joints. We can look at the inflammation in the joints. We can look at the cells that come into the joints. We can measure the inflammatory response, et cetera, et cetera. And so the basis of this model that we looked at was we took our mice that had the receptor, so they have this MICL, this inhibitory receptor, and mice that we generated that didn't have this receptor. And what we found was quite remarkable. So shown here in these full black circles are the mice that have the receptor. And you can see, and this has been published before, that in these what we call wild type mice with the receptor, you can induce the inflammation, and you get inflammation, and then over time it starts to resolve. But what we found in the mice that lacked this receptor was quite profound, that these mice, the inflammation was uncontrolled and it didn't resolve. So that's sort of, I guess you could understand that if this is a molecule, if Mickel is a molecule that regulates inflammation, you can see that this regulation is now gone in these mice. So this is playing a really important role in regulating inflammation in these animals. And we could show that using some really cool technology, and I'm just showing this because it is cool technology. So this is in vivo imaging, so we can actually image in live animals what's going on. And we've got two dyes here, one that osteosense that binds to essentially to bones. And you can see a very nice signal here in the wild type mice. But in the knockout mice, because there's massive arthritis and there's massive turnover of all the bones and inflammation in the bones, you can see that dye's gone. So that's an indication of bone remodeling, as it's called, and that shows you that there's something happening in the arthritis in animals. And we can also measure the inflammation directly. So this is a dye that you get this as the signal when there's more inflammation in the joints, and you can see the knockout, as you can see from this curve, is more inflammation. So the question then is, well, what's going on in these animals? What is Mickel doing in these animals that is driving this response? So we induced the response and we looked in the joints. I'm not going to go into any detail. Immunologists love to show these sort of plots, but the take-home message from this was we saw a specific type of immune cell called a neutrophil was much higher in the joints of the mice that were knocked out from Mickel than in the wild type mice. And the important thing about neutrophils, these are very early cells that respond to infection and danger or damage to self-insult. These are fantastic cells for controlling infection and responding to infection, but they're also very pro-inflammatory. They're full of granules that help kill pathogens, but these cells can also drive damage, and they are very good at killing things and causing damage both to pathogens and to our own cells. And what we found is that not only were these cells, excuse me, well present in the joints, but they were more activated using a variety of markers that allow us to measure that. So to show that actual fact that this cell type, this immune cell called a neutrophil was really key, we took the same mouse model, which I've already shown you now. And what we did was we did this whole arthritis model, and then we just took the neutrophils out of the mouse altogether and said, well, what happens when we take the neutrophils away? Does that do anything? And you can see, so this is your knockout here and your wild type, as I've shown you already. Knockouts have more severe disease. You take away the neutrophils, shown here at the bottom. In both diseases, it completely ablates the disease. So yes, the neutrophils in this model are absolutely responsible for the disease in both the wild types and the knockouts. It's something to do with the neutrophils. And can we prove that another way? So one of the things we can do, which is really cool, is we can take out the neutrophils. Let me go back a slide here. We can take the neutrophils out of this mouse, out of the knockout mouse. We can put them back into the wild type and say, well, is it the neutrophils themselves that are the problem? And that's the experiment. So we took neutrophils from the knockout mouse, we put them into normal mice. And when we did that, what we could see was even just the transfer of these neutrophils themselves caused more disease in the wild type animals. And you could see that both from clinical score here, but also actually if you looked at the tissue and you looked for damage and a variety of markers, you can see that down here. So what that told us was that actually it was this receptor expression on these specific immune cells that was absolutely critical in this model. So what is it about this receptor on these cells? What is the function of this receptor on these cells that's going wrong? So the neutrophils are, as I mentioned, are very good at killing things, and they're very good at causing damage. And one of the ways they do that is when they're stimulated by pathogens or danger molecules, damage from yourself, they can activate a process called the respiratory burst, which leads to the production of reactive oxygen species. So this is reactive oxygen components that react with things and damage them and kill them. And there was a paper some years ago by Jürgen Ruhlen's group that suggested that Mickel could indeed regulate this respiratory burst, the production of this really toxic pathway. And we could recapitulate that and show that indeed, yes, so this is just measuring the respiratory burst, the formation of these toxic compounds. We could see versus a control, let's ignore the control. You can see that when you have your knockouts, there's a massive induction of this respiratory burst. Now, this respiratory burst is present in neutrophils. It's very important, as I said, for killing pathogens, can also cause damage. But one of the things that's very specific about this response also in neutrophils is it can cause neutrophils to undergo what is known as neutrophil extracellular trap formation, which when I first heard about this, when it was published by Arturo Zielinski many years ago, I didn't believe it at all. I thought it was completely counterintuitive. So essentially, what a neutrophil extracellular trap is, it's a form of programmed cell death that neutrophils undergo. And essentially, they sort of, this is programmed, so it's engineered and organized by the cell. They die. And what they do, as you can see here on the right-hand side, is they die and they extrude all their genetic contents, their DNA, and all those granules of all their toxic proteins, and they pop them outside of the cell. Now, that to me sounded absolutely crazy because that's incredibly inflammatory. It drives massive inflammatory responses. Yet it's also really important for controlling infection. And the reason why it's so important for controlling infection is really exemplified in this picture here. You can see these bacteria are trapped by this trap. So they're very important for controlling infection, but they're also very important for driving autoimmunity. And so we wondered whether this respiratory burst that's been controlled by Mickel in the neutrophils was involved in controlling the formation of these neutrophil extracellular traps. And that's exactly what we found. I'm looking at the time. I'm going to need to speed up a little bit. You can see that the neutrophil extracellular traps much higher. Just look at this picture. You can see there's much more neutrophil extracellular traps being formed in the Mickel knockouts than in the wild types. And that's quantified over here. And when we went and looked back at our mice in their joints during this arthritis score, we could see the presence of these neutrophil extracellular traps using a variety of markers, but quantitated down here. So what this is telling us is that what Mickel is doing is it's controlling this respiratory burst, it's controlling this neutrophil extracellular trap formation, and the loss of this receptor is causing this to happen, induce a exacerbated response. And we could prove that because there's a very nice pharmaceutical inhibitor of the enzymes involved in driving these neutrophil extracellular traps. And so we did the same sort of experiment as I've shown you before, but now added the inhibitor and really just compare these white circles that you can see over here to the black circles. And essentially what it's showing you is that when you add the inhibitor to the knockouts, this can completely control the exacerbated inflammation that I'd shown you before. So what Mickel is doing is that it's playing a role in preventing excessive inflammation, preventing the formation of this very inflammatory respiratory burst and the formation of neutrophil extracellular traps. And when this receptor is gone, this control is absent, and then you get this massive increase in inflammation and damage to the joints. Now, one of the things we always try and do in my laboratory is we always try and correlate what we discover in mouse models to humans. And I'm sure all of you are aware of rheumatoid arthritis in humans, really nasty, debilitating condition. We had shown that the human receptor functions similarly to this receptor and that it could drive cellular inhibition. And what we were expecting to see was that we would have, as we'd found for many other molecules of immune function, that we'd find genetic changes that correlated with disease. And we looked very extensively. There are very big cohorts of patients that allow us to study genetic changes. We never found any correlation with any changes in rheumatoid arthritis, which was very disappointing at the time, as you can imagine. But one of the characteristics of diseases like rheumatoid arthritis and other autoimmune conditions is that these patients develop what are known as autoantibodies. So they develop antibodies that instead of targeting things like pathogens like COVID, they develop antibodies that target themselves and have been implicated in the pathology of those diseases. And when we looked in these patients, we found that indeed these patients had much higher levels of autoantibodies directed against this receptor than healthy control patients. And when we looked at a very large cohort of these patients to see if we could see any correlation between disease severity and the presence of these antibodies, we found a direct correlation between the marker of disease severity and the level of antibodies. So the patients who had more antibodies against this receptor had higher levels of disease. And what's interesting, just as a point of note, is that this disease measure actually measures is an indication of neutrophil extracellular traps. So we've got a very nice correlation between disease, neutrophil extracellular traps, and the level of these autoantibodies. Now, in our mouse models, if we take a mouse and we add autoantibodies to this receptor, we can recapitulate this disease. And we could also show in vitro that if we added antibodies from these patients to neutrophils, we could get increased respiratory burst, just like we saw in the knockouts, and increased production of the neutrophil extracellular traps. So in humans, very much similar to what I just showed you in mice, these autoantibodies are blocking this receptor and then driving this increased ROS production, increased net production that leads to exacerbated disease in rheumatoid arthritis patients. Now, the million-dollar question here is exactly what is this receptor recognizing? So I told you that these receptors, they recognize a ligand and they trigger intracellular signaling, and activation receptors like Tectin-1. That ligand, for example, is beta-glucan. But Mickel is also a receptor. It has to be recognizing something. And what is it that this receptor is recognizing? And what we discovered actually was that this receptor was actually recognizing the nets themselves. And so if you make a net and then you add it to cells from the wild type or knockout mice, you can see that the cells recognize the nets and they respond much more highly in the absence of Mickel, both in terms of inducing ROS and undergoing nets themselves. And we could show, and I won't go into the detail of this, but using a variety of approaches, we could show that this receptor was directly able to recognize the DNA components of these nets, which we could validate in an experiment. I won't go into that experiment now. So essentially what we have discovered is that what this receptor is doing is when these cells expressing this receptor go into this joint, they're actually recognizing these neutrophil extracellular traps. And this is then acting like a brake to prevent the induction or excessive induction of the respiratory burst, the neutrophil extracellular traps. But when you have antibodies to this receptor or you lose this receptor in our knockout mice, this control is gone. You get increased inflammation. And this actually leads to a pro-inflammatory feedback loop that then leads to the exacerbated disease we see in our rheumatoid arthritis patients and in our mouse models. Now, as I mentioned to you when I introduced the neutrophil extracellular traps, they are really important in controlling infection. And that's been shown for a variety of pathogens. But of course, as I mentioned, I'm interested in fungal pathogens. And we've shown or previously people like Konstantin Urban have shown that nets are really important in controlling infection. So does this Mickel have a role in controlling infection? And indeed, that's what we saw in vitro. So if we take our cells from the wild type and knockout mouse and we add now fungi to these cells, we can see that again, we get more increased neutrophil extracellular trap production, more increased ROS production. This occurs both in the mouse knockouts, but also if we block the receptor with autoantibodies. So what happens then if you infect these animals with this pathogen? So we did this experiment. We took our wild type and our knockout mice and we infected them with Aspergillus. And the result for me was absolutely staggering. So if you look here, this is the wild types in the black circles and the knockouts in the Mickel. So in contrast to the rheumatoid arthritis model, where you had much more severe disease in the Mickel knockouts versus the wild type, now if you have this loss of control of net formation, the Mickel mice are significantly protected from infection because they have more net formation, which allows them to control the disease better. And you can see that if you look at the fungal burdens in the tissues, for example, the brain, when you have uncontrolled net formation, the brain controls the infection and also the inflammatory response that we measure here. And of course, we can validate this response because we can come in with our inhibitor that blocks the net formation of nets. And if we do that, then we can revert the protection that we saw in the knockouts into the knockouts by inhibiting this receptor. In other words, making them as susceptible as the wild types to the infection. And that's also validated in the brain. Sorry, I'm going a bit quickly because I'm conscious of time. So then just to summarize what I've shown you. So what we essentially have discovered is that Mickel, through recognizing these DNA in these extracellular traps, blocks inflammatory signaling and controls this inflammation, which is really important to balance the ability of our immune systems to control infection, but at the same time to reduce autoimmunity. So this receptor is acting like a handbrake on the immune system. It's really triggering the balance between control of infection and the development of inflammation that can be damaging to our cells. But in the absence of this receptor, as I've shown in our knockouts, or if you're a patient with autoimmune disease that has antibodies to this receptor, this handbrake is missing, leading to more inflammatory signaling and an enhancement of an auto-inflammatory response that just drives more and more inflammation that's really damaging. And in this case, drives more exacerbated autoimmunity, but at the same time increases your protection from infectious disease. So what I showed you was rheumatoid arthritis patients. Of course, we think this is a universal mechanism regulating the balance of inflammation in our immune systems. And so we've started looking at a whole variety of other autoimmune diseases. So the two that we've looked at is lupus patients, patients who have lupus. Lupus patients are characterized by excessive net formation. Again, in the lupus patients versus our healthy controls, you can see that there are much higher levels of autoantibodies to this receptor. And very interestingly, COVID patients or patients who have severe COVID are also characterized by having increased net formation in their lungs. And in these patients too, we have discovered that that there's a much higher level of autoantibodies to this receptor that's correlated with driving inflammation in these patients. And in both of these patient groups, and also in the arthritis patients, if you take the autoantibodies directly from these patients, that's exactly what it does in vitro. It drives this enhanced inflammatory response, which is detrimental. The other aspect about of this is that this suggests that there could be a potential therapy and control of this exacerbated inflammatory response in these autoimmune patients if we could find a way of blocking this interaction between the autoantibodies and this receptor. And this is work that's ongoing in my lab. But the other thing, the other aspect about this discovery is that if we're able to block this receptor using antibodies, for example, we can help treat patients who are suffering from invasive fungal infections and other infectious diseases. And we've actually started to model that. I'll just show you one piece of data now just to prove the point. So now what we do is we take animals and we treat them with these autoantibodies and infect them to see whether these autoantibodies can drive this exacerbated net formation that would protect against infectious diseases. And that's exactly what we see. So if we take the animals and we add these antibodies, we can start to protect these animals from infection. So the patients who are suffering from severe invasive fungal infections, this could be a potential therapy, and we're busy looking at trying to translate that into the clinic, both in terms of using the antibodies to protect against fungal infection, but conversely trying to find ways of blocking this in autoimmune patients to protect against immunity. So that brings me to the end of my my presentation, and I hope I've shown you a little bit about understanding how our immune systems recognize and control infection, why I'm so fascinated by the balance of the functions of these receptors, how interesting they are, how good they can be, how bad they can be, and how devastating. And then finally, just to thank all the people in my group. So the star of the story that I mentioned was published just last week in Nature. Really, it was Mariana Malamud. He was the guy who really made the fundamental breakthroughs for us there, but also background work from Pierre and Lauren and all the other people and collaborators, specifically the people who provided clinical samples and clinical input into all of these studies. And if there are any questions and time for questions, thank you very much. Thank you very much for giving me the opportunity to present.
Thank you, Gordon, for an amazing talk. Really well explained for those of us who are not in your field and therefore finding it complicated. We do have time for a few questions, so let me ask the audience if anybody would like to ask a question. Please feel free to put your Zoom hand up, or perhaps just speak if you would like. If you can't find a hand, it's not always easy. Anyone like to ask? There's
a message in the chat, Stephanie.
I see that. Karen, would you like to speak and ask your question? Karen, sorry, it's Karen.
Hi, can you hear me, Stephanie?
Yes, hi, we can. Hi. Karen, go ahead.
I'm actually a Karen.
Oh,
Zimbabwean Karen. I have to be careful when I say that. So yeah, I'm an ecologist, Gordon. So thank you. That was really quite fascinating, and I'm not sure I entirely followed everything, but it really you explained it so well. And my question is really, perhaps maybe from an ecological perspective, is, can those receptors, can one control the activation of the receptors by external or internal factors other than, sort of you talk about therapeutic ways of doing that, but yes, for example, temperature or diet in the case of the mouse or the model or the human.
So we can control receptors and the impacts with drugs. So pharmaceuticals, yes. Our diets definitely have an impact on the way the receptors function, but that's a much more complicated answer because it involves our microbiomes and the interaction of our microbiomes with our immune system. So it's difficult to say if one specific factor can control that. Certainly things like compounds that we eat, for example, in the brassicas like broccoli and other things like that, can impact cellular responses again, but that impacts the microbiome. Temperature is a good one. I don't know the answer to that question, I'm afraid. I don't know if anyone's ever looked to see whether temperature has an impact on cellular responses. Because our temperature is homeostatically controlled, you'd only really think about that in terms of our external extremities, and I don't know the answer to that question. I do know that when people, we study mice, we put them at room temperature. I know that when people put mice at 37 degrees, which is their internal, there is changes in the way they respond. But in terms of the actual receptors, I don't know. But the one thing about temperature that I will say, which I didn't mention when I was introducing fungal pathogens, is that climate change one of the reasons that we are resistant to fungal infections, other than when our immune system is compromised, is because we're endotherm, so we have a high body temperature. And there's a very good argument by a colleague and friend of mine called Arturo Casadevall, who says that the reason why the dinosaurs went extinct after the big asteroid impact was because there were a lot of fungal spores kicked up in the environment, and that killed cold-blooded creatures, and whereas warm-blooded creatures can survive fungal infections. And a good example of that is the bats, the hibernating bats. Their body temperature drops, they get this white-nosed bat syndrome that's wiping out bat populations around the world. But the global warming that we're having is now increasing the activation of fungal virulence factors, and we're seeing the emergence of new pathogens. And there's a bug by the name of Candida auris. I don't know if you've ever heard of that. Candida auris has been found simultaneously, all of a sudden, in 4 continents. It's not present in the historical record. It's extremely drug resistant. It's thought to have evolved because of climate change into a human pathogen. And if it gets into hospitals, they can't kill it because it's resistant to most hospital detergents. It's closed down intensive care units in many hospitals. They have very strict practices. So the emergence of the environment on fungal pathogens is huge, which is not what you asked me, and I'm sorry, but I used that as an opportunity today.
But very fascinating. Thank you. Dean Goldring, you have a question?
Oh, thanks very much, Gordon. That was a absolutely fascinating talk, and I really loved some of your pictures and diagrams that made it very easy somewhat easier to follow what you were saying. These receptors, are they distributed amongst other cell types other than neutrophils, firstly? And secondly, the levels of these receptors, do they change in different disease states on your neutrophils? So
the receptors have, immune receptors in general are expressed differentially on different cell types. Things like the toll-like receptors are found on many cell types, but not all the tolls are there. And the same with the lectins. So you find some lectins on some cells, but not others. And your second question was, so this MECL that I told you about is expressed primarily on immune cells, the macrophages and neutrophils. You don't see it, for example, on stromal cells like epithelial cells or things of your blood or your tissues. It's really only seems to be very strictly limited to immune cells. And yes, the expression levels can change. So our inflammatory response, the cytokine that produce, can alter the expression of these receptors quite dramatically, actually. We don't really understand why that is. Sometimes it's counterintuitive. So, for example, dectin-1 I told you is really important for controlling fungal infections, yet one of the cytokines that induces its expression most highly is a cytokine that's associated with susceptibility to disease. So why does it do that? No idea.
Any other questions?
And can
Go
I
ahead.
follow up with another question?
Sure.
And are there other disease states that you see where a fungal infection assists with the disease that you've started to look for or found?
Assist, yes. Co-infections, I think, is what you're asking me about. I don't know of a single fungal infection as an infection that assists the disease. They are generally really bad. So there's been a really recent acknowledgement that influenza and COVID-19 lung infections, if you have a co-infection with a fungal pathogen called Aspergillus, which in actual fact we're breathing all the time, it increases your rate of mortality by something like 3 to 5-fold. So it's a really bad co-infection. TB and fungal pathogens are thought to have co-infections, other viral infections. We know very little about co-infections. Can fungi, not infection, can fungi assist? Yes. So we have commensal fungi that are thought to help train and drive our immune response in a positive manner, like in the microbiome. In actual fact, compared to the rest of the microbiome, we have very little understanding of how these organisms have a role. Fungi have also been associated with some very detrimental things like cancer. So there's some very clear evidence that certain fungal pathogens directly associate with the development of cancers, for example. Even alcoholism, which, if you want me to go into detail, is quite interesting. But yeah, so there's a whole lot of things, and there's talk now, but no evidence, strict evidence for it, that might be involved in things like Alzheimer's disease. So a lot of the fungi that we get, like Cryptococcus, which I'm pretty sure everyone on this call has got cryptococcal things that are in their brains that have stopped. Only if you get HIV will you get this in brain infection. But there's a possibility that that could be seeding some of the aggregates, the amyloid aggregates that drive Alzheimer's. That's just very speculative, but there's work going in that area, that fungi might be behind a lot of the pathologies in human, and some benefits too.
Thanks very much.
Thank you. If I might ask a very ignorant question, my biochemistry is engineering biochemistry, but I heard and listened to your speaking about therapeutics or therapies, but you never used the word vaccine.
Yeah.
And I just wondered, in situations where it's these very well-known fungal infections, and indeed some of the more modern, like the Candida auris that we do know about, is there a chance for vaccines? Is that going to be a possibility?
Yes. So there's not a single vaccine for human fungal pathogen in clinical use at the moment. It definitely can work. We know from our mouse model lots and lots of, but the field is so small, Stephanie, that's the problem. We have so few people working. So in my center in Exeter, we've got 17 principal investigators. That represents 40% of the entire UK community working on fungal pathogens, right? You look at bacteria, I mean, there's hundreds and hundreds of people.
Yeah.
In South Africa, there are a few spotted investigators. There are 4 in Cape Town in the Africa unit. I mean, it's so small. So the amount of work going towards that is very little. People have developed, so for example, for vaginal candidiasis, there's a vaccine that's been developed, it's been tested therapeutically up to phase 2 clinical trials. It's shown promise, but then no one invests in it. There's just not enough look at. So we are in the center, we're trying to get some more interest in the vaccine initiative to get it going. But yes, we could get vaccines and we could employ them, but there's just none and not enough people to do it.
Thank you. So of those who are in the audience, anybody like to ask anything else? I think we could probably go on forever, but we have used up our time. So Gordon, let me say thank you very much indeed, as my colleagues have said, for a really informative and very accessible talk on a topic which most of us are frightened of because it can be so very complicated. So thank you very much for your time, for your sharing with us your work, and we hope that it will go from strength to strength. So thank you very much indeed for your talk, and thank you everybody who has been with us this evening. And enjoy the rest of your evening. Thank you.
Bye-bye then. Thank you.